there is something present inside the nucleus which is acidic. There is something that acts as the transforming principle which carries the genetic information. Now, everything lies inside the nucleus. Now, how to prove it what it is? Because the nucleus contains DNA, it contains ribosomes, it contains RNA, it contains many proteins. And the strongest contenders for the genetic material candidature were the proteins and DNA. Now, how it is to be established that whether it is DNA or it is the protein? It took almost 10 years, and we have the biochemical characterization of Griffith's transforming principle. Now, the transforming principle is the one which is carrying the genetic information that was proved by Griffith. Now, what this transforming principle is, that was proved by Avery et al. There were 3 people, Avery, MacLeod and McCarthy. 3 of them individually studied that. No, not individually. They collectively studied during 1933 to 1944 that the biochemical nature of this transforming principle tells us that it is nothing but DNA, and it's not the proteins, it's not the other genetic material, that is RNA. Now, how to cut out the probability of these two? It is very simple. The entire transforming principle experiment is to be carried out, but every time you have to put a specific digesting enzyme. You have 3 types of enzymes, that is DNases, you have proteases, you have RNases. Now, how this is to be established that it is the DNA which is the genetic material. Now, we are going to revise what was in the transforming principle experiment. There was mice, there was S strain, and the mice got killed, right? There was mice, there was R strain, mice lives. There was mice, there was heat killed S, mice lives. There was mice, there was heat killed S plus R, right? And the mice gets killed. Now, this entire experiment is to be repeated, but in one case you have to use RNases. Now, RNases would digest the RNA of the S colonied cells, and the experiment would be taking place as it took place earlier. All these things would happen, or you can simply do this step, that in this step you add RNases, that RNases are not able to digest the transforming principle, and hence the mice get killed. Now, put proteases. When proteases were put, then also the transforming principle was not digested and the mice got killed. But when DNases, those enzymes which digested the DNA, then the mice lives. That means the information carrying particle has been digested, and what enzymes when used digested it? Those were the DNases. That means those enzymes which could digest the DNA could stop this experiment or could change the outcome as expected. That means the transforming principle got digested, and who digested it? DNases. We know that enzymes are very specific. That means it would be DNA which would be carrying the information, and because of the activity of DNases, that information got shattered, and hence the experiment did not complete as expected. So, it was proven that the biochemical characterization makes it clear that it is the DNA which is the genetic material and not the close contender protein or not RNA. So, this was all about biochemical characterization of the transforming principle which Griffith has proposed to be DNA.